It's my absolute pleasure to introduce Arnaud Sentis. So Arnaud's research examines individual, population, and community responses to climate change using manipulative experiments, ecological informatics, and theoretical models. So in ecology, such researchers are really rare. Many of us are good with experiments or mathematical modeling, but usually not both. And Arnaud has trained with leading researchers from all over the world. So he received his Bachelor's of Science from the University of Montreal and Aix-Marseille University. He received his Master's of Science in Ecology from the University of Quebec, and his PhD in ecology was a joint degree from the University of Montreal in Canada and Toulouse University in France. He was then a postdoc at the University of South Bohemia in the Czech Republic, the University of Toulouse in France, and the French National Center for Scientific Research. He is now a junior researcher at the National Institute of Agricultural Research in Aix-en-Provence, France. He has a long list of accomplishments for such a young scientist. He has published 31 papers in the top journals in ecology, including multiple papers in Ecology Letters, Global Change Biology, Ecology, and Heredity. He has successfully garnered funding for his research, and in 2015, he also received the Young Researcher Award from the French Society of Ecology. I met Arnaud as part of an international working group of scientists that studied species interactions in food web ecology, but I knew his research long before I met him. Indeed, I covered many of his papers in my classes as essential reading for any student that wants to understand how climate change affects communities and ecosystems. So it's my great pleasure to welcome Arnaud, and I'll let him take it away from here.
So, hello everyone, thank you for being here. I'm excited to present some of the research I did in the last several years. So I tried to put together some of the different pieces I did during my PhD, my postdocs, and briefly talk about what I'm doing now. So as Gina said, I'm working mainly with climate change and with temperature, and I try to understand the symptoms, acclimation, adaptation to thermal stress, and I'm particularly interested into species interactions. So I will start my talk with a question of my question is, are you stressed? So usually it's a better question when we are facing each other, but so I usually ask to raise your hand if you are stressed, but I will maybe see you not see you all, but yeah, so I have one hand here, even more hands, yeah. So do you think that stress is good or bad? So if you think that stress is bad, please raise your hand again. I can see the response. Okay, so a lot of positive stress, some negative stress. Okay, so here is the answer from the American Institute of Stress. So if the stress level is too low, then you are inactive and laid back. And when stress increases, there is an optimum level of stress for performance. And then when the stress is too high, then you have exhaustion, anxiety, and breakdown. So this shows you that the level of stress will really affect the performance in different ways. So I think with temperature, you often get this kind of curve where you have an optimal temperature. And then when the thermal stress is too high, you have a decrease and a breakdown of the different molecular processes or the behaviors of the organism. So you have different sources of stress. The stress can be abiotic, such as temperature. It can be biotic, or both at the same time. So I also try to look at interaction between stressors. So you have different symptoms. So the stress can affect the physiology, the behavior, and the phenotype of the organism. It can also affect interaction. So when you are too stressed or not stressed enough, you can feed more or less. And I think you can have the same kind of effect with temperature. So there are different ways to adapt to rapid environmental change. One way is to disperse. So this has been like widely reported, for instance, for butterfly species that are like moving northwards to track like optimal thermal conditions. You also have like phenotypic plasticity. So one of the important phenotypic response to temperature is a decrease of body size with temperature for many ectoderm species, both in aquatic and terrestrial systems. And then you also have microevolution based on selection from the stressors. But like many studies report that it's not that widespread and too slow in many cases, especially for species with long generation times. So I think like most evidence from adaptation to climate change come from species with short generation times. So another level of complexity is that species interact. So here you have like a trophic chain with 3 species, and of course temperature will affect the 3 species and their interactions. So we have knowledge of like how temperature will affect each of the species, but we have less knowledge about like how temperature will affect interactions. So in my work, I try to like include all these different aspects. So in terms of phenotypic response, I work mainly on phenotypic plasticity and not that much on dispersion, but this is also something interesting. So today I will try to present like some of the aspects of my work that cover these different questions. So I have 3 parts. The first part is on symptoms. The second part is acclimation. And the third part is adaptation to temperature stress. So I start with symptoms, and then at the end of this first part, you can stop me and I can answer questions. So this is like a bit more serious figure showing the effect of temperature. So you can see the same kind of like shaped curve that I showed before with the stress level. And so to describe the rising part of this curve, we can use the metabolic theory of ecology. So here you have a small equation. I don't know if you see my mouse. Can you see it? Yes? Okay. So here it says that the metabolic rate, or actually any kind of biological rate, increase exponentially with temperature. It also depends on the body mass of the species. So we can use this theory to describe the rising part of this curve here. And according to this theory, the slope of the temperature relationship should be like almost equal for all rates, and with an activation energy, so that's the value of the slope, that should be 0.65. So we can use this theory to try to make some predictions about species interactions. And this is what I did during my PhD. So I'm just showing here a lizard and a fly, but I didn't work on this anyways. So the temperature will affect both the predator and the prey. And using the metabolic theory of ecology, we can predict that the search rate, so the search efficiency of the predator to find the prey, will increase exponentially with temperature. And the handling time, so the time it takes to handle and digest the prey, will decrease exponentially with temperature. And based on that, we can predict what will be the effect of temperature on the functional response. So the functional response is the relationship between prey density and the number of prey eaten by a predator. So if temperature is low, then the search rate is low and the handling time is long. So it means that the search efficiency at low prey density is low, and it takes some time to handle the prey, so the maximum predation rate is also low. And when you increase temperature, then you increase the search efficiency, and you also increase the maximum number of prey eaten per unit of time. So we wanted to try these predictions using a simple experimental system with aphids and lady beetles. So we had an experiment lasting for 25 hours. We introduced different aphid density on these pepper plants. We had 6 different temperatures, And after 24 hours, we removed the predators and recorded the number of prey alive to get the number of prey eaten. So these are the results. So on the x-axis, you have the aphid density. On the y-axis, the number of aphid eaten for the 6 different temperatures. So you can see this plateau shape that we get. And from this different curve, we can extract the attack rate and the handling time, like using some nonlinear regression methods. And this is what we get for each of the temperatures. So this is the handling time here. This is the test temperature. So each dot come from one curve. And you can see that here we fit the model of the metabolic theory of ecology, and we can see that the handling time is decreasing exponentially with temperature, and the activation energy is not that far from the prediction. In this case, the metabolic theory of ecology works nicely. In the other case, so for the search rate, we can see an increase in search rate with temperature, but then at the warmer temperature, it starts to decrease. So in this case, the metabolic theory of ecology was not able to predict what would happen at higher temperature. And in our case, the search rate is driven by behavior. And what we observe is that the lady beetle, instead of hunting for aphids, they tend to like find cooler places and stay there and wait for the cold weather to come back. Okay, so if we use this information on search rate and handling time, what we can produce is a unshaped relationship between temperature and predation rate, whereas there is no predation at low and high temperature, and there is an optimal temperature for predation. So we can use this information to predict what could be the impact of climate warming on predation rate. So if the actual temperature is here, so much below the optimal temperature, then a +4 degree should lead to an increase in predation rate. So this would be a positive effect. But if the species is already close to the optimal temperature, then increasing by 4 degrees should lead to a negative effect of feeding rate. So the position of the environmental temperature compared to the optimal temperature for the trait is quite important. So this was like in a really simple trophic chain, but of course, so here I'm showing this picture where you can see that in nature you have like many species interacting together, and you often have like many plant species competing together, herbivores and predators. And we know that we have emergent effects from all these interacting species. So the question is like, can we translate what we learn from this like simple trophic chains to more complex food webs? So we can try to understand the effect on the whole food webs, but this might be complicated, especially in experiments. So instead, we can focus on trophic modules that are subsets of food webs, of species that tend to interact more among each other than with the other species in the web. And for this, I will show a simple example of like 3 species: a man, a rabbit, and a carrot. So when the 3 interact together, you have an omnivory model. But then if the man is not eating rabbits, and I think in the US, most people don't eat rabbits, but we do. So you have a competition between the rabbit and the man for the carrots. If the man is eating only rabbits, which is also possible, then you have a food chain. And in the place where the man is eating the rabbits or the carrots, you have polyphagy. So you can see like with the 3 species, you can have really different kind of models with only one link changing here. So the question we wanted to ask is what is the effect of temperature on these trophic modules? So here we used again the same system with lady beetles and aphid, but we introduced a 3rd species, which is a midge feeding on the aphid. but the lady beetle is also feeding on the midge. So we have an omnivory module where this is also called intragueal predation, where the intragueal predator is feeding on the intragueal prey and the extragueal prey. So now we have the effect of temperature on all the species, and we want to try to understand what is the effect of temperature on interactions. So we use, again, like functional response modeling to predict what should be the effect of temperature and prey density on predation strength. So that's the number of aphids eaten by the lady beetle. And we try to predict the strength of omnivory, which is this arrow here, which is the number of midge eaten by the lady beetle. So again, with temperature, we observe that, I think, yeah, this is not temperature. 100 degrees starts to be really warm. So I mismatched the label of the axis. But with temperature, you can see that omnivory increase and then decrease. And when you increase the enrichment, which is like the number of aphids in the system, then you have a dilution effect and you tend to have less omnivory. So then we decided to test the prediction. So we used again the same kind of experimental system, an experiment that lasted for 24 hours, 3 aphid densities and 3 different temperatures. And after 24 hours, we recorded the number of aphids alive and if we found the midge or not. So in gray, you have the prediction of our model with like confidence intervals. And in white, you have the observation for 10 aphids in the system, 45 aphids and 90 aphids. And you have the 3 temperatures, so 14, 22, and 30. So you can see here that we have a really good match between the model prediction and the observation. So again, we find that when you increase temperature, you tend to increase predation rate. And when you increase aphid density, you also have an increased predation rate. So for the omnivory level, so this is the number of midge eaten. So you have a good match between prediction and observation, especially at 10 aphids and 45 aphids. But here, when the model predicts that the number should be low, we didn't observe the interaction. So sometimes you are supposed to observe a little bit of predation, but we didn't find this. But what I find more interesting is that, so if I reconstruct this trophic module based on the number of prey eaten by the lady beetle and the number of prey eaten by the midge, which I didn't show, is that the shape of the module is changing with temperature and the number of aphids in the system. So here you have competition, this is competition. Here you have omnivory, omnivory. Here we have polyphagy. Here we have omnivory, omnivory, competition, and omnivory. So this is showing you that the shape of the trophic module is changing very quickly according to temperature and prey density. So the message, the take-home message is that food web structure is not static, but depend on environmental factors. So you could see that, you could think about like seasonal change in temperature, maybe even daily change and change in resource availability that can also shape the structure of the food webs. Okay, so I'm done with the first part. I can take a break here and ask questions. Not ask, but answer. Okay, so if there are no questions, I can continue.
No, I have a question.
Yeah.
Hi, thanks. So, a couple of questions. One, I was wondering if the temperatures in your experiment were constant or fluctuating, and if they were fluctuating, how high, like how much did they fluctuate, and
So
have you looked at that?
This experiment was with constant temperature. We also like did similar experiment with heat waves, so trying some fluctuations. And what was interesting with the heat wave is that we found that the presence of the predator buffered the effect of the heat wave on the aphids. So basically, the idea is that without predator, there is a strong effect mortality on the mortality rate of aphids. And when you introduce a predator, the main driver of mortality becomes the predator, and then the heat wave, they have a weaker effect on the aphids. Yeah.
And so the other question I wanted to ask is, there this idea or there's been evidence to show that species are closer to their optimal ranges in tropical versus temperate versus polar systems. So I'm wondering if you've thought about how those predictions of the shifts in the food web might play out differently and kind of different at different latitudes.
Yeah, this might be interesting to look at. Yeah. So I think again we expect that closer to the tropics, as species are closer to their optimum, you may have stronger change in food webs, but yeah, but I'm not sure if someone tested that, so that might be worth looking at it. Yeah.
Great, thanks.
Yeah, you're welcome. So I will continue with the. So what we did in this first experiment was that we had like the predator and prey at one temperature, and then we expose them to another temperature, and we look at the impact of temperature. But what we can see from climate change is that the increase in temperature is a gradual process. So what we did is what I call a sunburn experiment, where you place the organism at like a really different temperature. So what you may measure is a sunburn effect, is not like the long-term effect of temperature. So if you are more used to temperature and get tan, then you don't get the sunburn, right? So the idea here was to look at acclimation, and if acclimation can change the impact of temperature on trophic interactions. So to test these ideas, we used a different system with Daphnia and dragonflies. So we went, this was in Czech Republic, we went to a nice little pond, we collected some dragonfly larvae, and then we kept them in the lab for a while. So the design was that we split up the small dragonfly larvae into a warm treatment, so a +4 treatment, and the ambient temperature at which we collected the larvae. And then we maintained them until they reached the last larval instar at these two temperatures. And then we tested them in a cross-factorial design at two other temperatures. So the idea was to know like if this acclimation during that development will affect predation rates and metabolic rate. So the idea was to look at the energetic efficiency of the predator, which can be seen as like a feeding rate is the energy income, and metabolic rate is the energetic outcome. So again, we did like some functional response experiments where we take the ambient acclimated predator and the warm acclimated predator, and we measure the functional response at 17.5 degrees and 21.5 degrees. So we had different prey density, and we recorded the number of prey eaten. And from this, we extracted like this parameter, attack rate and handling time. So what we can see already here is that the warm acclimated predator, they tend to feed more, especially when they are at 21.5. I will show it to you with these parameters. So here you have again handling time, and here you have ambient or warm acclimated predator at the different test temperature. And what we can see is that handling time decrease with both the acclimation temperature. So if you compare the letters, and it also decreases with test temperature. So if you compare the numbers here. So basically the warm acclimated predator at the highest temperature, they had the shortest handling time. So they were able to handle a lot of prey per unit of time. For the search rate, we didn't find an effect of the experimental temperature, but there was an effect of the acclimation temperature, where warm acclimated predator were less efficient at finding the prey than the cold acclimated predator. So here, so I am jumping back to the figure I showed before. So I was showing that this +4 degree could lead to a negative effect on predation rate. But the idea is that it could lead to a positive effect of predation rate if the warm acclimated predator have if the thermal performance curve is the position of the thermal performance curve is at a higher temperature. So this is for the metabolic rate. So we like introduced the larvae in some like glass chamber where we recorded the oxygen, and from oxygen consumption we estimated metabolic rate. So this is the metabolic rate in joule per hour, and you can see that it increased with the experimental temperature as it should, but there is no effect of acclimation on metabolic rate. So there was only an effect on predation rate. So then, using these results of predation rate and metabolic rate, we can estimate the energetic efficiency, which is the balance between predation and metabolic losses. And what we found along a gradient of prey density is that the warm acclimated predators that are in red, they have a higher energetic efficiency. So they tend to perform better when they are acclimated to warm temperature. And experimental increase also, experimental warming also increase their energetic efficiency. It means that for this species, probably that the optimum temperature is higher than this 17.5, and that the warming treatment that we did improved the fitness of the species, the organism. So to conclude this part, so we had an effect of thermal acclimation on predation rate, on the predator energetic efficiency, and we also use some population dynamic model to look at the long-term interaction strength, which is an estimate of the stability of the system. And what we found is that the thermal acclimation tend to decrease long-term interaction strength, so it has a stabilizing effect on this system. So the conclusion from that is that acclimation may rescue predator from extinction and stabilize food webs as it increased the energetic efficiency. Okay, to continue with acclimation, so I was showing before that the body size of many species tend to decrease with warming, so they get smaller when it's warmer. And a lot of people have been working on why it should be so. So there have been different hypotheses on like dissolved oxygen, metabolism and respiration, and our developmental rate can influence the size of species. But there is much less research on the ecological consequences of this smaller size with warming. So here we wanted to test that. What are the consequences of being smaller in a warmer environment? So when I'm coming back to this picture, I'm thinking about the direct effects, so the effect on physiology and interactions, and the indirect effect mediated by changes in body size. So for the direct effect, we can use the metabolic theory with some limits, as I showed. So for the indirect effect, so there have been different hypotheses to explain and models to explain why a species gets smaller with warming. So one explanation is that the thermal dependency for growth rate is lower than the thermal dependency for developmental rate. So basically, they both grow faster and develop faster, but they develop faster than they grow. So they reach a smaller size at maturity. But so this is single model, but when you look at meta-analysis on how body size change with temperature, you can see that the pattern is really variable, and you can see that even some species get bigger when it's warmer. So you can see that this single model cannot really explain this diversity of patterns. And we observe this both in marine, freshwater, and terrestrial systems. So with David Bukal and INRAE Binzer, we use a food chain model. So it's a Volta Maltera model, where you have a top species feeding on the intermediate species, feeding on the basal species. So here you have the logistic growth equation for the basal species. Here's the functional response of the intermediate consumer. He has a loss with the mortality, with the metabolic rate, and a loss with the feeding of the top predator in the system. So here we, in the model we use, the parameters, they depend on temperature and the species body mass. So we use different meta-analyses that analyze the effect of temperature and species body mass on each of these parameters. And we use this general relationship that have been like mainly estimated for insects to parameterize the model and try to test some realistic scenarios. And for the extra parameter in the model is that the body mass of the species, they also depend on temperature. And for the parameterization of this equation, we use the paper I was showing before, the meta-analysis by Foster et al. So this is just, you don't have to look at the equation, but this is just to show the temperature dependency of the different parameters. So we have carrying capacity that is decreasing with warming, the growth rate that increases with warming, metabolic rate increasing with warming, search rate has this shaped curve that I was showing before, and handling time which decreasing with warming. So for the body mass function, so as I was saying before, the strength of this slope is very variable from one species to another. And you can see it here in this meta-analysis. And another level of complexity is that the slope depends on the body mass of the species at a given temperature, so at 20 degrees. So basically, for small unicellular organisms, they all have the same slope. For aquatic ones, the larger they are, the steeper is the slope. And for terrestrial one is the opposite. So it might be a bit complex to get, so I will explain this in more details. So for instance, for unicellular algae, when you increase temperature, whether the algae is large or small, they will have the same slope. So they will reduce in the same way with temperature. But for aquatic species, the larger they are, the steeper is the slope, so the smaller they will get. And for terrestrial ones, it's the opposite. So the larger they are, the shallower is the slope. And even for some large beetle species, they found that they tend to increase with temperature. So here we used a mix of these different slope scenarios. And in the model, we did some simulation, like in a range of temperature from 0 to 40. We varied the fertilization level from 0 to 20. And we also tried different body mass ratio because we know that trophic interactions are often size structured with predator larger than their prey. And then we have different temperature, size, rule, strength, so different slope for this body size change. So we tried the reverse one where they get like larger with warming. We tried the maximum reduction, so where they get really smaller with warming. And then we used the slope for terrestrial species and the slope for aquatic species. And we had two scenarios, one where all species change in the same way with temperature, and one where only one of the species responds to temperature, because we also know that from one species to another, it can differ. So these are the results from the simulation for one body mass ratio and no TSR, so they do not change with temperature here, their body mass. So here you have fertilization level and temperature. So in the middle, you can find a region where 3 species coexist. So the number of surviving species is 3, the orange area. Here they are in a stable equilibria, so which means that the density is not fluctuating. And here you have fluctuations. On the upper left corner, you have an area where you have only 2 species. So the top species get extinct at the warmer temperature. And then the intermediate species go extinct at even warmer temperature. So you end up here with only the basal species. And here with the fluctuation, what happens is that the fluctuation, they get too strong. And then you have the extinction of both the top predator and the intermediate predator. So you have a region here where there are like too many fluctuations when fertilization level increase. Well, this is known as the paradox of enrichment. So what we did is to look at the area of this orange area. So the percentage of system persistence. So the idea was to look at the region where the 3 species can coexist and how this body size change can influence the area of this persistence area. So what we saw is that when you have the TSR and a strong body mass ratio in the system, you have an increase of system persistence with the TSR. So basically, if they all get smaller with warming, then the size of this orange area will increase. So you will have a higher persistence of the system with the body size decrease. But what we also found is that this effect really depends on who's changing body size in this food chain. So I was showing the scenario where they all change, but you could see that if only the basal species is shrinking with warming, then you can get the opposite effect. Then if the intermediate change, you have this shaped effect. And if the top species change, you also have a kind of shaped effect. So it's really showing that depending on what the species are doing, like in the food chain, you can get different outcomes. Okay, so to conclude this part, so we saw that this body size reduction can increase the system persistence. But it also depends on who's changing in the food chain. And what we saw also is that this effect of body size reduction is stronger in size-structured ecosystem. So we could think that aquatic systems are typically size-structured compared to a terrestrial system. So the impact of this body size reduction, they might be stronger in aquatic system compared to terrestrial systems. So I'm done with this part, and then we'll start the last part of my talk. So if there are any questions on this second part.
I had a couple questions again.
Yeah.
So you showed a bunch of different responses, predicted responses to temperature. And I didn't get to see all of them, but one of them was that carrying capacity decreases with warming. And I was wondering, first of all, I wasn't sure which level of the food chain that was. And I just wasn't sure why that would be predicted, because I was thinking if they're getting smaller as it's getting warmer, wouldn't they be eating less per individual? So why doesn't the carrying capacity go up?
Yeah, so we used this. So I'll come back to show.
Sorry, I know that was a little bit before.
Yeah. So that was here. So the carrying capacity is affecting like the population density of the basal species here.
Mm-hmm.
And so we used from this study where they show that the carrying capacity exponentially decreased with temperature. But in the literature, there have been some people arguing that that this carrying capacity might not change with temperature or even increase with temperature. So we also tried that. I didn't show the results here, but what you find basically is that when you increase, so if carrying capacity is not affected by temperature or increase with temperature, you'll find that this extinction threshold here should happen at higher temperature. Because one effect of temperature is to decrease this carrying capacity here, to decrease the fertilization level effect, basically. So when you're at warmer temperature, you also have less resource in this model. So you will go extinct because of temperature and the low resource level. And if you increase resource level, then you'll increase the extinction, the thermal extinction threshold. Yeah.
Is that different for aquatic versus temperate systems too? Like they might be differentially affected by temperature when you're talking about like basal producers?
Yeah, I think so, because like plants, they have like much longer generation times compared to
Phytoplankton.
unicellular algae. And
Mhm.
also like with carbon, it's quite different. The quality is changing a lot also
Right,
with warmer temperature and CO2.
that's right.
And they have different compound, which you don't find much in aquatic systems. So
Mhm.
like many kind of things that can differ like between these two systems, which are interesting to compare also.
Mhm.
I think this kind of modeling approach based on body size will mainly work with aquatic systems.
Aquatic, yeah, where it's gape
Mhm.
limited and stuff like that. Yeah. Mhm.
That brings up another issue that I was thinking about that would be different in aquatic systems versus terrestrial systems, because warming, even though overall warming may be gradual and directional, you get a lot of fluctuations, and that gets back to the acclimatation issues. And so when you're thinking about these different stable states, have you looked at how things respond when you have like bigger fluctuations in temperature versus just an overall change in temperature? And is that different in different systems?
Yeah, that's a good point. So I didn't do it, but some people did. And actually they found that the influence of this temperature fluctuation can be stronger than the impact of just increasing the mean temperature. So definitively, I think this is something important to consider. And here we have like a really simple model, and the idea was more to inform us of like potential consequences than like trying to get accurate predictions of a given system. But yeah, I fully agree that these temperature fluctuations they are quite important. I could send you
Thank
some
you.
papers if you want.
Oh yeah, for sure.
Some other questions or? Okay, then I'll continue with the last part, which is like the link between acclimation and adaptation. So there is, I think, more and more interest into epigenetics and all this acclimation response. They can be transmitted from one generation to another. So just like to have a few definitions. So epigenetic is defined as the mechanism of non-genetic heredity, and so it could be like intra-organism among cells or like cross generations, but it includes all molecular process of variation in gene expression. So it includes methylated DNA, microRNA, histone modifications. And just to give one example is that methylated DNA regions explain 60 to 90% of heritability in the flowering time of Arabidopsis. And this epigenetic mark, they can be transmitted from one cell to another or from one generation to another. And also one interesting thing is that cytosine, when they are methylated, they are much more likely to mutate into thymine. So it means that this epigenetic factor, they can also increase the likelihood of mutation and then evolution by selection. So this raised the question of is this epigenetic heredity a first step toward genetic assimilations? So I'm giving you just an example of epigenetic heritability here is that the other fear conditioning in mice. So what they did here is that they stressed the mice with another and electric shock. So the mice, they learn like quite quickly that they should avoid this other. And then they look at the methylation pattern into the sperm cell of the male. And then they did some couples with a female and they raised their offspring. And then when they use the same udder, the offspring is feared. So as a stress reaction, but it was never like taught by his parents. So it was raised on his own. And this was transferred to the grand offspring. And what they found and what they show is that this methylation mark on the DNA, they were transferred from one generation to another, and they affect the genes that are responsible for the receptors of this particular other. So if they would change the other, then there would be a different genes that would be affected by this methylation pattern. That was, I found a nice example of this epigenetic heritability. So in this experiment, we used the aphid again. So aphids are cloned, they reproduce parthenogenetically. So here you could see a mother with a baby attached. And so we used this clone, and the idea, was to expose them to predators. And aphids usually, they don't have wings unless they are stressed. And one of the stress is predators. So when you introduce predator, then the mother, which is wingless, will produce daughter with wings, but they are all clones. And we know, like from the literature, that the development of wings is based on some methylation patterns and some hormonal regulations too. So in this project, we wanted to look at the importance of transgenerational phenotypic plasticity for evolution. So we had two stressors, predators and heat wave. And we wanted to look at phenotypic plasticity and its role for the fitness of the aphids, basically. And we also wanted to look at DNA change, so change in allele frequency, and a potential link between this DNA methylation and the mutation rate in the aphids. So what we did is that we started from a single aphid. We amplified the population, so we get a lot of aphids, and then we splitted them in 4 different treatments. So for each of the treatments, we had 10 replicated lines where we had aphid that we transform from one generation to another in absence of predators and heat wave. We had aphid with heat waves, with predators, sorry, aphid with only heat wave, and then aphid with lady beetles and heat waves. And we maintained them for 13 generations like this. So when we look at the proportion of winged aphids, so this is when we started the experiment. Then we started introducing the predators and the heat wave. This is the dotted line, the dashed line, sorry. And what you could see is that without heat wave and without predators, so that's the red line, then we have around like 20% of winged aphid in our population, which is like the kind of standard for this clone. When we had heat wave, we had a lower production of winged aphids. But when we introduced predator, we first had this really high peak in proportion of winged aphids. So around 80% of them become winged. And then it stabilized around like 50%. And you could see that here we could not see any difference between the heat wave treatment and the non-heat wave treatment. So there was an interaction between predators and heat wave on the proportion of winged aphids. So the question is like, are these phenotypic change stable? So what happened when you remove the stressors? So we wanted to look at this transgenerational heredity. So when you remove the stressors, you could see that suddenly the proportion of winged aphid, they jumped. So they become much lower, and then they come back to the level of the control without stress. But it takes like 3 generations to come back to the level of the control. So this has this like long-lasting effect for at least 3 generations. So the explanation here is why it becomes lower than the control is that the winged aphid, they tend to produce mainly unwinged aphids. So when you remove the stressor, then all the winged aphid, they produce mainly unwinged, so it becomes lower. And then the unwinged, they start producing wings or winged aphids again, so you come back to the level of the control. So we continued the experiment until like 27 generation, but without the heat wave, because this was like too much work. We had only like the 2 treatments with and without predators. So these are the results until generation 27. So you can see that we maintain this like level of 50% of winged aphids in presence of predator. And then the question is this plastic response stable? So we did like 3 parallel experiments where we removed the predators. So again, it becomes lower than the control and then came back. And it took like 4 generations to come back to the level of the control. Then we did that a second time a bit later. And it also took like 4 generations to come back to the control. And then we did that a 3rd time, and then it was taking 5 generations to come back to the level of the control. But here, so when we did some analysis to estimate like the generation time at which they should come back to the level of the control, using this only the black and blue curves. And what we found is that the longer we expose them to predators, the longer it takes to come back to the level of the control. So basically, as there is a kind of memory and imprinting, and the longer you expose them and the longer they will remind that they were exposed to predators. So the question is, did evolution start to occur or not really? So we didn't continue the experiment, so we don't have the answer. Ouais.
Non, c'est pas grave. Fais gaffe à l'heure, hein.
Ouais, ça marche. They're chasing me away because it's time to go out. I'm okay. I still have a bit of time. And I'm almost done, so okay.
Janet, do you have a question on what?
Yeah, I guess maybe, I think you answered it, but it was how long, so in the earlier experiments, was it just one exposure to the stressor for just one generation, and then you got the effect through time, or was it?
It was continuous. Yeah, in the first experiment, so we here.
In that one, yeah.
All this was with continuous exposure, and here we removed, like we stopped the heat wave and we removed the predators.
I see. So, okay. But then they're just continually being exposed to the same, I guess I'm not understanding. So they're just being exposed to the same stressor.
Yeah. Yeah, that's why we wanted to remove the stressor, just to know if it was like a phenotypic plastic response that was like reproduce every generation, or if there was something else going on, like a beginning of assimilation.
I see. And okay. And then in the last part, how is that different? Sort of the one where you showed how the more
It's just that,
the exposure.
yeah, okay. So this is generation 13 where we,
Mhm.
so actually the first removal experiment is the same results I was showing before, except that not showing the heat wave, right?
Mhm.
This is exactly oh no, sorry. This is the same, yeah.
Mhm.
And so we did 3 times the same experiment of removing the predators. So
I
this,
see. And they would,
the line here was maintained with predators. And then we took like half of the aphids from these cages and we removed the predators and we maintained them like for 1, 2, 3, 4, 5 generations.
I see. Okay. All right. I get it. And then the last one is just it just kind of linearly scales with
Yeah.
how long.
Yeah, we use some polynomial regression to have a better estimate of the exact generation time when it comes back to the control.
Okay. All right. I get it. Thank you.
Hitting some curve here, like because you can see that there is some variation. So we did like some bit more advanced statistical methods to get these 3 points, which corresponds to these 3 blue lines coming back to the level of the black line.
Right. Okay. Thank you.
Yeah, you're welcome. Yeah, so we did, then at the end, we did a common gathered experiment to compare the fitness and the plasticity of the line. So we used the control line in black and we used this blue line where predator has been removed for 3 generations. So we used again like the same kind of design where we had this treatment without predator and a treatment with predator. So we had these 2 evolutionary treatments, the blue and black, and we had these 2 test treatments with and without predators. So what we found is that the one that evolve with predator, they tend to have a weaker response to predation, so they produce less winged aphid in presence of predator than the one that evolved without predators. So basically, this match with the finding that we have that the longer we expose them to predator, the less they produce winged aphid. And so we explain this in our system by the fact that, in fact, the winged aphid, they cannot escape because they are in a cage and they have a lower fecundity. So we have a counter selection against the winged aphid. So if I had to do this again, I would maybe have a design where the winged aphid could escape and they would probably have a better fitness. So we have a kind of particular selection where particular situation where we have counter selection against the winged aphid. And in terms of population growth rate, which can be seen as a measure of fitness, what we found is that the aphid lineage that evolved without predators, they have the same fitness as the one that evolved without predator in absence of predator. But when you expose them to predator, they have a higher fitness. And this is probably explained by the fact they have less winged aphid in their population. And so they have a higher fecundity. So here we show that there is trait variation, trait heritability. I didn't show, we did another experiment to measure that, and fitness implication. So there is evolution by selection that is possible based on this epigenetic variability. So we have a decrease in the percentage of winged aphid. So there was a selection against winged aphid in our system. And the number of generations to come back to the control increased. And there was a decrease of plasticity under constant predation pressure. Yeah. So the general conclusion is that, so you have this direct effect of temperature. You have phenotypic response or phenotypic plasticity can interact with this direct effect and modify this direct effect of temperature. And then some potential link with this like short or midterm phenotypic response with longer term in terms of with longer time scale with evolution adaptation. Yes. And then this can feed back to the direct effect of temperature, of course. So I'm just like one slide of what I'm doing now, right now. So I have two main projects. One is to look at the impact of reduced body size on aquatic systems. So we have this mesocosm here in Aix-en-Provence, and we have fish lineage that have been maintained for like 9 years at 2 different temperatures. So we have like smaller fish at warmer temperature, and we want to introduce them in the mesocosm, and we can also control the temperature. So we will look at the impact on the food webs in the small mesocosm. And we also have a survey of French lakes where we have data on fishes, macroinvertebrates, and plankton. And we can reconstruct the food webs from these different lakes. And we want to see which environmental which environmental factors are the driver of the food web structure along this gradient of lakes. And with that, I would like to thank all the people that have been involved in all these different projects. And I would also like to thank all the funding for this different projects. And finally, I thank you for your attention. And if you have more questions, I'll be happy to reply.
Thank you so much, Arnaud. Do folks have any other questions for Arnaud? He's 6 hours ahead, so we just need to be a little mindful of his dinner time.
Can I ask you one question, Arnaud?
Sure.
I really liked your epigenetic story at the end, well, partially because I teach a course in epigenetics and it's really cool. You probably haven't yet uncovered the mechanism to account for the persistence of the phenotype after you removed the pressure and what has changed? Do you have a loci in mind that are under control?
Yeah, so we wanted to do some sequencing of the methylation mark, but at the end, so the sad story is that I finished my postdoc there and then I left and no one took over. So the aphids are still in the freezer and, and no one sequenced that. So we don't know actually the mechanism, but that would be interesting to look at.
No, because it's, so you probably are better read than I am on the literature, but there's not very many examples going cross generations. I mean, there's many at the cellular level. So at the cellular level, that's easy. But from multiple generations, there are some plant examples. I don't know of very many animal examples that are, essentially going through meiosis, and that's really stunning. So
yeah, there are a few examples with C. elegans, I think, and but that's true that there are not that many demonstrations of this epigenetic.
Yeah, it could be a really big story, I think, if you could figure out the mechanism that underlays it. Is it a microRNA or is it something else that's regulating this? What is resetting it after 3 to 5 generations?
I was discussing with some people in Rennes who are doing that, like looking really at the mechanism, because I'm not a specialist, and but they were saying that with the experiment we did, because we are working kind of at the population level, and the sample of aphids that we have is a mix of like different stages and things like this, then they argued that if you want to recover the mechanism, then you need like to have like really some experiment design to collect the aphids that could go for the sequencing, because the stage, the state, the density, everything, like a lot of different things can affect the methylation pattern. And we may have too much noise to find like the signature of this predation pattern on the methylation mark. But yeah, so that was also one of the reasons why we did not try to sequence all the aphids that we have in the freezer. But maybe one day we'll do.
Yeah, maybe if the sequencing costs go down, it'll be easier.
Yeah.
Arnaud, do your aphids have the Buchnera endosymbionts
Yeah.
that help them with the heat shock? That's because there could be some like microbial interaction going on there as well.
Yeah. So they have many different symbionts. I don't think Buchnera is helping, but there is another one which is helping. I forgot the name, but which is helping with the heat shocks. Yeah.
Mm-hmm. Mm-hmm.
Yeah, that was also something we discussed at that time, and we wanted to have a look because as I was presenting before, so the generation times of these like smaller organism or symbionts, they are much shorter, so you can have evolution
Much faster, right?
in the symbiont than in the aphid itself. So.
Well, I mean, people have to really appreciate the size of these experiments that you're conducting. They're a nightmare. I can hardly think of how you kept all that going and kept sane at the same time. So like doing a bunch of like Buchnera stuff on the side would be difficult. So
yeah, that was intense.
Mm-hmm. Do folks have other questions? You're getting a bunch of thank yous and great talk in the chat too.
Okay.
This just
Thank
covered absolutely everything. It's nice because we have a really diverse department, and you're getting many mercies. But anyhow, we have a really diverse department, and I feel like you touched on something for everybody, like a little bit of neurobiology, a little bit of evolution, a lot of ecology. So there was something for everybody here. So all right, so I'll let you get to dinner here, Arnaud, but I just wanted to
thank
tell
you.
Thank you so much again for staying with us. I know you're 6 hours ahead, and for letting us really cut into your day to talk to you. But in a way, I'm so happy that we can do a Zoom departmental seminar like this, because now I can get some of my wonderful European colleagues to talk. So that's fantastic. So I just really appreciate you making time for us. So.
Yeah, thanks so much. That was great.
You're welcome. It's nice, very nice to show my work to other people too.
Thank you so much, Arnaud.
Yeah.
All right, take good care of yourself.
Thanks.
Bye.